Apo B48. Apo B48 is a remnant of A. RNA editing, B. RNA interference, C. RNA splicing, D. DNA translocation. In the liver cell, ApoB100 ApoB100 gene gives rise to ApoB100 protein. But ApoB48 protein is made in intestinal cells from the gene encoding ApoB100 by the process of RNA editing, which is also known as differential RNA processing or chemical modification of RNA. This is a post transcriptional modification. RNA splicing is also a post transcriptional modification, but this is removal of introns and not editing. RNA interference is silencing technique where the gene is silenced by not letting the product of gene form. Translocation occurs during translation. This movement of ribosome over mRNA. So the correct answer here is A. RNA editing. Hepcidin decreases iron absorption by inhibiting hepcidin decreases iron absorption by inhibiting A. Divalent metal transporter, B. Hepastin, C. Ferroportin, D. Transferrin. Hepcidin regulates iron metabolism. This we all know. It mainly regulates iron entry into circulation. It mainly regulates iron entry into circulation. By inhibiting ferroportin, hepcidin reduces dietary iron absorption. The iron release from macrophage is also reduced by ferroportin inhibition. So increased hepcidin activity is responsible for reduced iron availability. Increased hepcidin activity is responsible for reduced iron availability. Increased hepcidin will lead to anemia. Increased hepcidin will lead to anemia. Decreased hepcidin will lead to iron overload. Hepcidin synthesis and secretion by liver is controlled by iron stores within the macrophages, inflammation, hypoxia, erythropoiesis. Other options. Divalent metal transporter absorbs divalent metal ions from intestine, especially ferrous. Hepahestin involved in homeostasis of iron and copper. Transferrin is a protein which is responsible for transport of iron. So the correct option here is C. Ferroportin. Which one of the following has the highest glycemic index? Which one of the following has highest glycemic index? A. Glucose, B. Fructose, C. Sucrose, D. Sorbitol. Glycemic index. This is the increase in blood glucose after test dose of carbohydrate compared with that an equivalent amount of glucose. Glycemic index is a value assigned to foods based on how slowly or how quickly these food cause an increase in blood glucose level. The highest glycemic index is for glucose and galactose. Highest glycemic index is for glucose and galactose. Also lactose, maltose, thialose, isomaltose get converted to glucose. So they also have high glycemic index. Glycemic index of fructose is less than that of glucose. Fructose and sugar alcohols have less glycemic index because they are not absorbed completely. Sucrose also has low glycemic index than glucose as it gets split into glucose and fructose. So the correct option here is A. Glucose. GLUT5 is transporter of fructose. GLUT5 is for the transport of fructose. So the correct option here is fructose. Which of the following is insulin dependent? Only GLUT4 is insulin dependent. Insulin recruits GLUT4. Okay. GLUT4 is present on membranes of peripheral tissue, that is skeletal muscle, cardiac muscle, and adipose tissue. It helps in insulin dependent glucose uptake after the meals. So the correct option here is D, GLUT4. Cause of HHH syndrome, hyperornithemia, hyperammonemia, homocystinuria is A. Deficiency of ornithine transcarbamylase. B. Defect in ornithine transporter. C. Deficiency of ornithine aminotransferase. The correct option here is D. All of the above. Defect in ornithine transporter leads to HHH syndrome, hyperornithemia, hyperammonemia, homocystinuria. This prevents ornithine transport into mitochondria. Hence, if in the cytoplasm, that is hyperornithemia, it reduces ability to clear carbamyl phosphate and ammonia, that is hyperammonemia. Mitochondrial carbamyl phosphate carbamylase lysine to homocysteine, leading to homocystinuria. So the correct option here is defect in ornithine transporter. Which method is used to separate a mixture of lipids? It is chromatography. Wrinkled vapor appearance of macrophage in bone marrow spread it is seen in Gaucher's disease. In CRISP9 Cas9 system, which repair mechanism is used for genome editing? It is non-homologous end repair. CRISP-Cas9 endonuclease system introduces double-stranded breaks in DNA at the specific target site. Now cell tries to repair this double-stranded DNA break by two mechanisms: non-homologous end repair or homologous repair. Non-homologous mechanism is always a possibility as it does not require homologous DNA. So it is a major repair mechanism during CRISP-Cas system. So correct option here is non-homologous end repair. Chimeric DNA, a chimeric DNA is used for, correct option here is organ transplantation. Organ transplantation. Rappaport-Lubric cycle shifts oxygen distribution curve to right because of 2,3-DPG. 2,3-DPG. Heart flushes are experienced as a result of increased noradrenaline and decreased estrogen. Restriction fragment length polymorphism is used for. Restriction fragment length polymorphism or RFLP is used to identify a change in genetic sequence that occurs at a site where restriction enzyme cuts. So the correct option here is trace inheritance pattern, identify specific mutation, and for other molecular genetic techniques. Thermogenic food, which of the following? It does not depend on macronutrients. Ketone bodies are Not utilized by ketone bodies are not utilized by RBC. In RBC, there is no mitochondria and ketone body utilization takes place in mitochondria. So RBC cannot utilize ketone body. Brain, muscle, and heart can use ketone body during starvation. Enzyme deficient in hyperammonemia type 1 is carbamyl phosphate synthase 1. Hyperammonemia is a metabolic disturbance characterized by excess of ammonia in blood. It is a dangerous condition that may lead to brain injury and death. It may be primary or secondary. This usually occurs in urea cycle disorders. Type of mutation seen in sickle cell anemia is point mutation, which replacement of amino acid does not alter its normal function. Glutamine replaced by asparagine. The clinical picture given below is due to deficiency of correct option here is A tryptophan. Tryptophan. Which of the following binds mRNA with ribosome in eukaryotes? It is 7-methyl methylguanosine cap. In eukaryotic cells, ribosome recognize most eukaryotic mRNA by binding to 7-methylguanosine cap at 5' terminus. Shine-Dalgarno sequence (SD sequence) is present at 10 position of mRNA only in prokaryotes and is rich in purine. In smaller subunit of ribosome, 16S RNA is present. SD sequence is complementary to 16S RNA and their binding helps in binding of mRNA with ribosome during initiation of translation in prokaryotes. Which of the following is an anaplerotic reaction? Conversion of pyruvate to oxaloacetate. Anaplerotic reaction are those reactions which synthesize TC and nitrates. Conversion of pyruvate to oxaloacetate is most important anaplerotic reaction. Which of the following group of protein assist in folding of other proteins? It is chaperones. Hmm. Myoglobin has which structure? Primary, secondary, tertiary. Best investigation of metabolic disorder is tandem mass spectroscopy. Separation of protein by mass is gel filtration chromatography. You can join my 6-day masterclass by clicking the link in the description box. You can also ask for any doubts from on the given number, WhatsApp number. Kindly WhatsApp. Please WhatsApp, don't call.